Hi, I am Sohail. Today I am going to talk about endoplasmic reticulum. The main points of the lesson are structure and composition of endoplasmic reticulum and types of endoplasmic reticulum, that is rough endoplasmic reticulum and smooth endoplasmic reticulum. Endoplasmic reticulum is a large dynamic structure that serves many roles in cell, including calcium storage, protein synthesis and lipid metabolism. Endoplasmic reticulum having a surface area up to 30 times than that of plasma membrane. Endoplasmic reticulum is called membrane factory of the cell, where membranes are produced and assembled. There are different factories in a cell, like nucleolus is known as factory of ribosomes. Ribosomes are known as protein factories. Mitochondria is known as powerhouse of the cell, and endoplasmic reticulum that is known as membrane factory. Endoplasmic reticulum is literally the network within the cytoplasm. The structure of endoplasmic reticulum is a network of membranes that is called cisternae. So, the membranes of endoplasmic reticulum that is called cisternae, singular is cisterna. It is an extensive system of membranes running throughout the cytoplasm. Endoplasmic reticulum, it has two surfaces, that is cytosolic surface and luminal surface. They contain different sets of enzymes. Now, what is cytosolic surface and what is luminal surface? Cytosolic surface is the outer surface of endoplasmic reticulum that is facing the cytosol, that is called cytosolic surface. And luminal surface is the inner surface of endoplasmic reticulum that is facing the lumen, that is called luminal surface. Cytosolic surface and luminal surface, they have different set of enzymes. As we can see in this diagram, ribosomes are attached to cytosolic surface and the luminal surface is smooth. So, ribosomes, they are not present in luminal surface. Ribosomes, they are only present here in the cytosolic surface. Endoplasmic reticulum that acts like a factory with different regions, and these regions, they have different synthetic capabilities for a particular product. Enzymes are located within the endoplasmic reticulum lumen. So, different sets of enzymes are present in cytosolic surface and luminal surface. What is the structure of membrane of endoplasmic reticulum? Endoplasmic reticulum membrane, which is phospholipid bilayer, that is embedded with extrinsic and intrinsic protein that also contains glycoproteins, proteins with carbohydrate attached, and glycolipids, lipid with carbohydrate attached, and cholesterol molecules are also present in the membrane. As we can see in this slide, nucleus is bounded by nuclear membrane. Nuclear membrane is a double membrane, that is outer nuclear membrane and inner nuclear membrane. There are certain points where outer and inner nuclear membranes are continuous. At these points, nuclear pores are present, and outer nuclear membrane, it is further continue attached to the endoplasmic reticulum. There are two types of endoplasmic reticulum, that is rough endoplasmic reticulum and smooth endoplasmic reticulum. Rough endoplasmic reticulum, they contain ribosomes which are attached to the cytosolic surface, and smooth endoplasmic reticulum, they are without ribosomes, so their outer surface is smooth. Rough endoplasmic reticulum that is studded with ribosomes that appear as dark granules. The lumen site of rough endoplasmic reticulum that appears bare, mean without ribosomes, whereas the outer surface, which is called cytosolic surface, that looks rough because of the ribosomes. Nuclear envelope is one of the functional domain of rough endoplasmic reticulum. Why? Because the outer and inner nuclear membrane, they are actually continuous with endoplasmic reticulum. That is why the nuclear membrane is one of the functional domain of rough endoplasmic reticulum. And in case of rough endoplasmic reticulum, ribosomes are attached to the cytosolic surface, as well as the ribosomes are also attached to the outer nuclear membrane. So, this is enlarged structure where we can see that outer and inner nuclear membrane, they are continuous at this point, which is called nuclear pore, and then outer nuclear membrane, it is further continuous into this structure which is called here rough endoplasmic reticulum. This is another slide where we can see inner nuclear membrane and outer nuclear membrane, they are continuous at this point where the nuclear pore is present. And outer nuclear membrane, it is continuous into this structure which is called rough endoplasmic reticulum. And ribosomes, they are attached to cytosolic surface of rough endoplasmic reticulum, as well as the ribosomes are also attached to outer nuclear membrane. Ribosomes, they are present on outer cytosolic surface of nuclear envelope, but ribosomes, they are not attached to nucleoplasmic surface. So, this is the inner nuclear membrane where the nucleoplasm is present. So, this is nucleoplasmic surface where the ribosomes are not attached, but the ribosomes are attached to outer nuclear membrane. But rough endoplasmic reticulum, they are involved in the synthesis of protein, and that protein is exported from the cell as digestive enzymes, proteins destined for the compartments of other organelles, and this protein might be integral part of the membrane. Integral part of the membrane means it is very important, and membrane cannot function without protein. What is the process of protein synthesis? Protein synthesis, it consists of transcription and translation. Transcription takes place inside the nucleus, that is the synthesis of messenger RNA from DNA, and translation that takes place in cytoplasm. When messenger RNA that enters into the cytoplasm, where small and large subunits of ribosomes, they are present. So, small subunit that binds with messenger RNA, later on the large subunit binds with small subunit to form a ribosome. Now, that ribosome reads the genetic information from messenger RNA, which is in the form of codon, and that information is translated in terms of polypeptide chain, which is synthesized on the surface of the ribosome. And in this polypeptide chain, number of amino acids, type of amino acids, and sequence of amino acids is determined by the genetic information present in mRNA. So, transcription that takes place inside the nucleus and translation that takes place in cytoplasm. During translation, when ribosomes are attached with messenger RNA, now that ribosome binds with the cytosolic surface of endoplasmic reticulum, and they are involved in the synthesis of protein. Ribosomes, they synthesize a protein, that is a polypeptide chain, and this polypeptide chain, it passes through a tunnel within the ribosomes into an endoplasmic reticulum pore, and that polypeptide chain, which is protein, that passes directly into endoplasmic reticulum lumen. An integral membrane protein is a type of membrane protein that is permanently attached to biological membrane, and proteins that partially passes through ER pore before being integrated into the membrane lipid bilayer. In the ER lumen, proteins are assembled and may be modified by enzymes that add carbohydrates or lipids to them. Carbohydrates can be attached to proteins to form glycoproteins, and carbohydrates can be attached to lipids to form glycolipids. Glycolipids and glycoproteins are structural component of biological membrane. Destruction of defective protein. Proteins that are not processed correctly are transported back to cytosol, and in cytosol there are structures which are called proteasomes. They are involved in the degradation of that defective protein. Proteasomes are protein complexes which degrade unneeded or damaged proteins by proteolysis. Proteolysis is a chemical reaction that breaks peptide bonds, and enzymes that help such reactions, they are called proteases. So, properly processed proteins that are to be routed to other compartments within the cell. Proteins are packed in small transport vesicles that bud off the endoplasmic reticulum membrane. Here we can see these small vesicles, they bud off from endoplasmic reticulum. Rough endoplasmic reticulum can therefore be considered as cell's membrane factory because they are involved in the synthesis of protein. And we will see that smooth endoplasmic reticulum, they are involved in the formation of phospholipid. So, phospholipid, protein molecules, glycoproteins, glycolipids, all these structures are synthesized by endoplasmic reticulum. That is why it is called membrane factory. Pancreas is an organ that is located in abdomen, and pancreas has dual function. It has endocrine glands and exocrine glands. Exocrine glands that produces digestive enzymes, and endocrine glands, they are involved in the production of protein hormones. So in this slide we can see exocrine glands of pancreas, they are involved in the production of enzymes which helps in digestion of food, and here are endocrinal tissues of pancreas which are called islets of Langerhans. Islets of Langerhans that produces insulin, glucagon, somatostatin and pancreatic polypeptide hormones are produced. So the cells in pancreas, they contain more proportion of rough endoplasmic reticulum because they are involved in the synthesis of enzymes as well as hormones. Rough endoplasmic reticulum, they also helps in repair and renew the membranes of the cell, and they also contribute to the formation of other organelles. Now, smooth endoplasmic reticulum. Smooth endoplasmic reticulum, they have tubular appearance and its membrane surface that appears smooth. Enzymes on the cytosolic surface. Cytosolic surface, as we know, the outer surface of endoplasmic reticulum that is facing the cytosol, that is called cytosolic surface. So the enzymes on the cytosolic surface of smooth endoplasmic reticulum, they are involved in the synthesis of lipids and carbohydrates. Smooth endoplasmic reticulum is the primary site for the synthesis of phospholipid, and phospholipid, one of important structural component in biological membrane, as well as they are also involved in the synthesis of cholesterol. In some cells, such as found in fat tissue, where smooth endoplasmic reticulum, they have enzymes that form lipid droplets. Smooth endoplasmic reticulum, they are also involved to synthesize steroid hormones, including the reproductive hormones from cholesterol. Steroid hormones, they are secreted by adrenal cortex, testes and ovaries, and all steroid hormones, they are derived from cholesterol. In liver cells, smooth endoplasmic reticulum, they play important role in enzymatically breaking down of glycogen. Glycogen is stored in muscles and liver. So in this way, they maintain the blood glucose. Amount of smooth endoplasmic reticulum is not same in all cells. That's variable and it depends on the function of the cells. So large amount of smooth endoplasmic reticulum that is present in liver cells, where they are involved in the synthesis of lipid. And the second important function of smooth endoplasmic reticulum is detoxification of toxic substances. So what is detoxification? Enzymes in smooth endoplasmic reticulum membrane of liver cells, they are involved in the breakdown of toxic chemicals. Then cell, they convert these compounds to water soluble products, and these water soluble products later on excreted from the body. Alcohol abuse that causes liver inflammation, because alcohol stimulate liver cells to produce additional smooth endoplasmic reticulum that increases the rate of detoxification. Another important function of smooth endoplasmic reticulum is they are involved in storing calcium ions. Calcium ions is a signal for the beginning of many cellular events like muscle contraction and glandular secretion. In muscle cells, specialized smooth endoplasmic reticulum are present, which are called sarcoplasmic reticulum. And sarcoplasmic reticulum, they store calcium ion which are needed to trigger, to initiate the process of muscle contraction. So in this muscle cell, we can see sarcoplasmic reticulum. These are modified smooth endoplasmic reticulum, and they store calcium ions, and these calcium ions, once they are released, they initiate the process of muscle contraction. The concentration of calcium ion in cytosol is kept low. When such events are not occurring, then calcium ions are pumped into endoplasmic reticulum. In the stomach of humans and other mammals, smooth endoplasmic reticulum, they are involved to produce hydrochloric acid. Let's watch this simulation.
The endoplasmic reticulum lies just outside of the nuclear envelope and is made up of a number of tubules and sacs called cisternae. Now these cisternae are made up of phospholipid membranes, and these membranes separate the internal compartment of the endoplasmic reticulum called the lumen from the cytosol of the cell. The membrane of the endoplasmic reticulum and the membrane of the nuclear envelope are continuous with each other, and as a result, the intermembranous space between the membranes of the envelope is also continuous with the lumen of the ER. And the ER is divided into two different sections, the smooth ER and the rough ER. The smooth ER, shown here, is responsible for synthesizing lipids, metabolizing carbohydrates, and detoxifying drugs and poisons. If you take a look down here, we'll illustrate how the smooth ER detoxifies. Under normal circumstances, anytime a drug or poison enters the body, the smooth ER will attach a hydroxide group that makes the poison more soluble in water and more easily excreted from the body. So if a person takes drugs or drinks alcohol excessively, this will induce the proliferation of the smooth ER so that the person can detoxify at a higher rate. The rough ER, shown here, is responsible to synthesize cellular membranes and modify proteins that are going to be secreted from the cell. Now the way that this works is that proteins are fed into the lumen of the ER by ribosomes that attach to its surface. As ribosomes translate mRNA, they feed the newly synthesized protein into the lumen of the ER, where it's then modified before it's sent off to the next organelle called the Golgi apparatus. Once the protein has been modified to a point in the ER, those proteins are packaged into a transport vesicle such as this, and those vesicles, which are simply small membranous sacs that bud off from the ER, are transported through the cytosol to the Golgi apparatus.
To sum up, endoplasmic reticulum, it's a dynamic structure that serves many roles in the cell, including calcium storage, protein synthesis, and lipid metabolism. There are two types of endoplasmic reticulum, that is smooth endoplasmic reticulum and rough endoplasmic reticulum. Ribosomes are attached to cytosolic surface of rough endoplasmic reticulum, but in case of smooth endoplasmic reticulum, the cytosolic surface is smooth. There is a checklist about the functions of endoplasmic reticulum, like they are involved in the synthesis of protein. IMP, that stands for integral membrane protein, which is synthesized by endoplasmic reticulum. They can add carbohydrates to proteins and lipids to form glycoproteins and glycolipids. Defective protein is degraded with the help of proteasomes in cytosol. Endoplasmic reticulum, it's a membrane factory where the biological membrane is synthesized. They are involved in the formation of digestive enzymes. Hormones are produced with the help of endoplasmic reticulum. Cholesterol is synthesized by smooth endoplasmic reticulum, and cholesterols, they are used as a precursor, starting material for the synthesis of steroid hormones. Smooth endoplasmic reticulum, they are also involved in the detoxification of toxic substances, and smooth endoplasmic reticulum, they store calcium ions. Let's watch this slideshow.